Okay, I took the video off with the hopes that some of the uploading and downloading times would be shorter, so you won't see my pretty face for this particular tutorial. This is going to be section 9.2 in chapter 9 on the neuromuscular junction we're coming to, but before we get to that, I wanted to take a moment and review the sarcoplasmic reticula inside the skeletal muscle fiber. So we note here in this diagram this yellow material here, kind of orangish yellow, a lot of little tubes. This is the sarcoplasmic reticulum. That's the singular term. Reticula would mean more than one. And you'll notice that this yellowish set of tubes in between the myofibrils is quite extensive. It extends in between all of those myofibrils. Now these larger yellow structures you're seeing are mitochondria, right? Remember from a previous chapter, these are the organelles that produce the ATP, right? That is going to be necessary for contraction and for relaxation. And so in addition to the abundant mitochondria, note again the extensive networking of the sarcoplasmic reticula, which you see here in cross sections, these little bitty oval, kind of orange oval structures. Those are these tubes that contain calcium ions. That's pretty much what this organelle does. It stores calcium. You'll note that in addition to the extensive networking of sarcoplasmic reticula between the myofibrils within the muscle fiber, on the surface of the cell membrane or sarcolemma, we have these little openings or pores, and these lead internally to form what are called transverse tubules, or T-tubules for short. They're called transverse because they extend inward at a 90-degree angle to the overall length of the fiber and myofibrils. That's where the term transverse comes in. And so again, they extend in between all of these myofibrils, the T-tubules do. On either side of the T-tubule, you see this enlarged, dilated structure. This is part of the sarcoplasmic reticula, and these are referred to as cisternae. They're just kind of enlarged, widened components here. We'll talk more about what they do in just a moment, but this is part of the sarcoplasmic reticula. And the T-tubule extends right between the two cisternae to make up what's called a triad. The triad. So here's a triad, here's one, here's one, here's one. There's a whole bunch of them. Okay, let's go on to the next slide. Here's the neuromuscular junction that I had mentioned earlier. This is literally where a motor neuron or nerve fiber, there are motor neurons within a nerve fiber, where they come down and come into close contact with the muscle fiber. So that's what neuromuscular refers to. Junction means kind of where they come together. So here we see a motor neuron. You might remember us talking about multipolar neurons a couple, 3-4 weeks ago. So this is going to send a motor impulse, basically a nerve impulse, down. And at the tip of this motor neuron, we see these sort of large pad-like structures. It's where the axon kind of terminates. And here we're looking at one such knob. So we're looking at one of these, blowing it up. And we see that this actual part of the motor neuron doesn't actually touch the cell membrane or sarcolemma. It gets awful close to it, but there's a little gap, or cleft it's called, between the folded sarcolemma. Okay, so the cell membrane is folded here. You'll see why it's folded in just a few minutes, as opposed to being nice and smooth. There's a gap between the end of the motor neuron, the axon of the motor neuron, and the folded sarcolemma of the muscle fiber. You'll note also inside the axon, there are organelles here called synaptic vesicles, and they contain what's referred to as a neurotransmitter. And the neurotransmitter, in this particular instance, has a special name. It's called acetylcholine, or ACH for short. That's the neurotransmitter found inside those little synaptic vesicles of the axon. And when an impulse comes down this axon, okay, and it gets to the end of the axon where these little kind of elevated or dilated little pads are, it's going to cause the synaptic vesicles to move toward that cleft like these guys have, and they're going to dump their neurotransmitter, acetylcholine, into the cleft. And here you can see these little blue dots. That's the acetylcholine, and it diffuses across the cleft, and it actually makes contact with special receptors on the surface of the folded sarcolemma. So I'm drawing these little kind of very poorly drawing these little green structures. Those are protein receptors that the acetylcholine will fit into, kind of like a lock and key. Okay. There's a video here that I'd like you to watch. I can't play it here on the voiceover PowerPoint, but if you go back to the regular PowerPoint of Chapter 9, you'll be able to play that video. There's also a good Khan Academy video that goes through this as well. But I do want you to go back and take a couple minutes and watch that at this point. Maybe stop the PowerPoint now, go back, watch that 3-4 minute video, and then come back to the PowerPoint. So I'm going to pause here for a minute. I'm going to pretend you've gone back and you've watched that, and then we'll pick up from there. Okay, I'm assuming you've watched the video, and it talked a little bit about how in response to that nerve impulse coming down the axon, action potential it's sometimes called, that calcium ions move in to the axon here of the motor neuron, and that calcium, that flooding in of the calcium, is really what propels these synaptic vesicles to actually move toward the cleft. Okay, it's the influx of calcium in response to that action potential or or nerve impulse coming down the axon. And then we've got the diffusion of the acetylcholine across the cleft. It, as we said, combines with special protein receptors. And now you may know the reason for the folded sarcolemma. The reason you have the folded cell membrane of the muscle fiber is that you can pack more of these acetylcholine receptors, right? If you've got that folded sarcolemma, you can put more of those receptors, and therefore more of that acetylcholine can combine with more receptors. And in response to that acetylcholine fitting into the receptor sites, that initiates what is referred to as depolarization of the sarcolemma. That's just sort of a fancy way of saying you're going from a nerve impulse now to the generation of what's called a muscle impulse. It's due to voltage changes that occur as a result of the sodium ions moving in from the cleft to the inside of that muscle cell membrane. The influx of the sodium induces the formation of the muscle impulse. And that muscle impulse then travels along the entire surface of the cell membrane of the muscle fiber, and it's going to go into those T-tubules and cause them to release their calcium into the myofibrils. And we'll talk more about that a little bit later. Here's another slide that shows one of those end little foot pads, if you will, of the motor neuron, where it comes into close association with the sarcolemma. Doesn't really touch it. There's that little cleft, remember, between the two. But here you can see one of those. There's another one up here in the upper right-hand corner up here. So these are branches coming off the axon of the motor neuron that comes into close association with the muscle fiber. And here you can see this is one muscle fiber here. There's another one up here, and there's another one down here. Well, we talked about the fact that the sarcolemma was really, really highly folded so that we could pack more of those acetylcholine receptors, right, on the sarcolemma. And this slide just kind of talks a little bit about how certain chemicals, and the one that we'll make brief mention of right here is a chemical derived from a tropical plant in the Amazon called curare, and the native indigenous people of the Amazon actually go out into the rainforest, or maybe they even grow this in their gardens. It's possible, I don't really know for sure, but they have access to this curare, and what they do is they take some of the secretions from the plant and they put it on their darts and their spears, their blow darts, and then they will shoot an animal, a monkey perhaps, or a tapir, which is kind of like a pig, and that curare will immobilize the animal, kind of paralyzes it. So what it really does, the curare is going to block those receptor sites we talked about. Remember a few minutes ago that ordinarily the acetylcholine would fit into these little protein receptors on the sarcolemma, but instead we've got curare coming in here and basically combining with those receptor sites. So I'll pretend this is curare in red. Here's curare. It blocks the receptor sites, so the blue acetylcholine can't fit in. And if the acetylcholine can't fit in, what won't be generated? You won't generate a muscle impulse. You won't depolarize that membrane of the sarcolemma, and you won't generate a muscle impulse. So the result is that the animal has no control over its skeletal muscle. It's basically paralyzed. It falls from the tree because it can't move. And then the individuals, get the animal, harvest it, and eat it. So that's kind of interesting.